Hello everyone, myself Vishal Tekade. I am studying in first year of BSc Microbiology at Fergusson College, Pune. My project topic is from genes to genetic diseases, what kinds of mutation occur. Let's see in short why did I choose this topic? Because I interested in the field of genetics because genetics helps explain a lot of things like what's make you unique. I make a career in field of genetics, so that's the reason I chose these topics. To study the genetic diseases, first we have to know what is gene. Gene is a unit of heredity. It contains the genetic information. It is made up of DNA. It is transmitted from the parents to the offspring. Gene unit of heredity information that occupies a fixed position means locus on a chromosome. Genes achieve their effect by directing the synthesis of protein. The term gene was given by the Danish botanist William Johnson in 1909 for any particle to which property of Mendelian factor or determinant can be given. Thomas Hunt Morgan in 1910 defined gene as any particle on the chromosome which can be separated from other particles by mutation or recombination is called as gene. In general, gene is the basic unit of inheritance. According to recent information, a gene is a segment of DNA which contains the information for one enzyme or one polypeptide chain coded in the language of nitrogenous bases or the nucleotides. The sequence of nucleotides in a DNA molecule representing one gene determines the sequence of amino acid in the polypeptide chain, means that is the genetic code. The sequence of three nucleotides reads for one amino acid means codon. Khorana was awarded Nobel Prize for the synthesis of an artificial gene. Gene acts by producing enzymes. Each gene in an organism produces a specific enzyme which controls a specific metabolic activity. It means each gene synthesizes a particular protein which acts as an enzyme and brings about an appropriate change. Gene is chemically DNA, but the length of DNA which constitutes a gene is controversial three terms, that is cistron, muton and recon. Cistron means it is a unit of function. Second one is muton. It is a unit of mutation. And third one is recon. It is a unit of recombination. These three terms were given by the Seymour Benzer to explain the relation between DNA length and the gene. Let we see about gene and its impact on our body due to mutation that leads to genetic disorder. Genetic disorder is a disease that is caused by a change or mutation in an individual DNA sequence. A genetic disorder is an illness caused by changes in a person's DNA. In 1983, a genetic marker linked to HD was found on chromosome 4, making it the first genetic disease to be mapped using DNA polymorphism. However, the gene was not finally isolated until 1993. In 1990, the first gene to be associated with increased susceptibility to familial breast and ovarian cancer was identified. Scientists had performed DNA linkage study on large family who show characteristics related to hereditary breast ovarian cancer syndrome. They named the gene they identified, which was located on chromosome 17, breast cancer gene 1. However, it was clear that not all the breast cancer families were linked to breast cancer gene 1, and with continued research a second gene, breast cancer gene 2, was located on chromosome 13. Everyone has two copies of both breast cancer gene 1 and breast cancer gene 2, which are tumor suppressor genes. If a person has one alternate copy of either gene, it can lead to an accumulation of mutations, which can then lead to the tumor formation. HD is inherited as an autosomal dominant disease. A gene mutation is a permanent alteration in the DNA sequence that makes up a gene such that the sequence differs from what is found in most people. Mutations range in size. They can affect anywhere from one single DNA building blocks means base pair to the large segment of a chromosome that includes multiple genes. Gene, muti- Gene mutations can be classified into two major ways: hereditary mutation and acquired mutation. Hereditary mutations are inherited from a parents to the parents and are present throughout a person's life in virtually every cell in the body. These mutations are also called germline mutation because they are present in the parent's egg or sperm cell, which are also called as germ cells. When an egg and a sperm cells unite, the resulting fertilized egg cells receive DNA from both parents. If this DNA has a mutation, the child that grows from the fertilized eggs will have the mutation in each of his or her cell. Then second one is acquired mutation, means somatic mutation. Occurs This occurs at some times during a person's lives and are present only in certain cells, not in every cell in the body. These changes can be caused by environmental factors such as ultraviolet radiation from suns, or can occur in an error is made as DNA copies itself during cell division. Acquired mutation in somatic cells cannot be passed through the next generation. Genetic changes that are described as de novo means new mutation can be either hereditary or somatic. In some cases, the mutations occur in a person's eggs or sperm cells but is not present in any of the person's other cells. In other cells, the mutation occur in the fertilized egg shortly after the egg and sperm cells unite. Means it is often impossible to tell exactly when a de novo mutation happens. De novo mutations means a new mutation. As a fertilized egg divides, each resulting cell in the growing embryo will have the mutation. De novo mutations may explain genetic disorder in which an affected child has a mutation in every cell in the body put the parents do not, and there is no family history of the disorder. Somatic mutation that happen in a single cells early in embryonic development can lead to the situation called mosaicism. Mosaicism. These genetic changes are not present in a parent's eggs or sperm cells or in the fertilized egg, but happen a bit later when the embryo includes several cells. As the cells divide during growth and development, cells that arise from the cell with the altered gene will have the mutation, while other cells will not. Depending on the mutation and how many cells are affected, mosaicism may or may not cause health problems. Most disease causing genes mutation are uncommon in the general population. However, other genetic changes occur more frequently. Genetic alterations that occur in more than 1% of the population are called as polymorphism. They are common enough to be considered a normal variation in the DNA. Polymorphisms are responsible for many of the normal differences between people, such as eye color, hair color, and blood types. Although may polymorphism have no negative effect on a person's health, some of these variations may influence the risks of developing certain disorders. It may be of different three types. First is frame shift mutation, second one is point mutation, and third one is duplication. The Human Genome Project has estimated that the human genome contains around 20,000 to 25 genes. Each of these genes is made up of hundreds to millions of DNA nucleotides. Sometimes only a single DNA mutation, means change in a DNA sequence, can cause a person to have a devastating genetic diseases. And researcher has been able to identify mutations responsible for causing thousands of different genetic diseases and the conditions. But sometimes a DNA mutations may do not harm at all. It all has to do with what the DNA mutation is and where it is located. Every gene in the human body consists of DNA, means deoxyribonucleic acid. DNA is a genetic code that is made up of four different types of nucleotides. First is adenine, second is guanine, third is thymine, and fourth is cytosine. The DNA code is turned into RNA, means ribonucleic acid, in our body in a process called transcription. In RNA, a nucleotides called uracils substitutes for every thymine. The RNA then goes through a process called translation to turn into amino acid. During translation, every three RNA nucleotides codes for a single amino acid. This sets of three nucleotides is called a codon, and different codons may code for the same amino acid. In the end, a sequence of DNA has been turned into a sequence of amino acid joined together in a long chain, which is called a protein. Proteins are responsible for more of the function of our cells. Many things can happen during this process to prevent a gene from turning into protein or to have a non-functional protein created. In a gene, if a single DNA nucleotide is mutated, for example from an adenine to guanine, this may cause the wrong amino acid to be made. If the wrong amino acid is made and assembled into a long chain of amino acid, the resulting protein may not work. This is because different amino acids are different in many ways, such as size and in the electric charges they have. These different characteristics affect the other molecules that surround them, means such as other amino acids and water. For example, positively and negatively charged molecules prefer to interact with each other and with water, which is called as being hydrophilic, whereas nonpolar molecules do not like to interact with charged molecules or with water, which is called being hydrophobic. This seemingly small difference can have very large consequences. For examples, the disease sickle cell anemia. Sickle cell anemia is one of a group of disorder known as sickle cell disease. Sickle cell anemia is an inherited red blood cell disorder in which there aren't enough healthy red blood cells to carry oxygen throughout your body. Normally, the flexible round red blood cells move easily through blood vessels. In sickle cell anemia, the red bloods are shaped like sickles or like moons. These rigid sticky cells can get stuck in small blood vessels, which can slow or block blood flow and oxygen to parts of the body. There are no cure for most people with sickle cell anemia, but treatment can relieve pain and help prevent complications associated with the disease. The symptoms and signs of the sickle cell anemia usually appear around 5 months of age. They vary from person to person and change over the time. Signs and symptoms can include anemia. Sickle cell anemia breaks apart easily and die, leaving you with too few red blood cells. Red blood cells usually live for about 120 days before they need to be replaced, but sickle cells usually die in 10 to 20 days, leaving a shortage of red blood cells means anemia. Sickle cell anemia is caused by a mutation in the gene that tells your body to make the iron rich compound that makes blood red and enables red blood cells to carry oxygen from your lungs throughout your body, hemoglobin. In sickle cell anemia, the abnormal hemoglobin causes red blood cells to become rigid, sticky, and misshapen. There is a risk factor for a baby to be born with sickle cell anemia. Both parents must carry a sickle cell gene. In the United States, US, sickle cell anemia most commonly affect black peoples. The mechanism of sickle cell anemia is when sickle hemoglobin give up its oxygen to the tissue, HbS stick together, forms long rods form inside RBC. RBC become rigid, inflexible and sickle shape, unable to squeeze through small blood vessels, instead block small blood vessels. Less oxygen to the tissue of body. RBC containing HbS have shorter lifespan, normally 120 days. And the second example of this disease is hemophilia. Hemophilia is an inherited bleeding disorder when blood doesn't clot properly. It is caused when blood does not have enough clotting factor. A clotting factor is a protein in the blood that controls bleeding. There are two types of hemophilia. Both have the same symptoms. Hemophilia A is the most common form and is caused by having reduced levels of clotting factor 8. Second one is hemophilia B, also known as Christmas disease, is caused by having reduced levels of clotting factor 9. So my project is completed. Thank you.